Okay, good afternoon. My name is Jessica Stark and I go to Union College, and I have the opportunity to participate in Hunter College's summer program for undergraduate research. And the title of my talk today is the role of AMPA receptor trafficking in the amygdala during Pavlovian conditioning. My project is focused on how we learn by associations. I'm interested in understanding what in the brain lets a rewarding association form, and also what in the brain lets us no longer make these associations. For example, if you hear an ice cream truck and you associate it with getting the reward of your favorite flavored ice cream, but one day you hear the ice cream truck and they tell you that they stopped making your favorite flavor of ice cream. Now you have to modify this rewarding association and know that when you hear the ice cream truck that you will no longer get that favorite flavored ice cream. Studying these associations is relevant because of their ability to translate to clinical issues, such as rewarding associations made in substance abuse, rewarding and aversive associations in eating disorders, and aversive associations in phobias. Our target then becomes something in the brain that creates the association between the stimuli for these items and the reward or aversion that comes from them. In understanding how these associations between stimulus and reward are made, we are then in a better position to know how we might be able to modify them, which would be particularly helpful for rewards that have negative impacts on our lives. Fortunately, we have a very simple way of modeling these associations through Pavlovian conditioning. For my project, we used an animal model of a rat where we presented a tone and a reward of food at the same time or paired so that the rat would learn to associate the tone with the food reward. Our first group of rats received only Pavlovian conditioning. A second group of rats went through Pavlovian conditioning and then an extinction protocol. The extinction protocol is used to explore whether this rewarding association can then be modified. To do this, we presented the same tone to the rats, but no longer paired the tone with food. Therefore, the rat had to modify the association made during Pavlovian conditioning to know that the tone no longer meant that the food was coming. So, my question is, are there distinct molecular mechanisms in the brain associated with Pavlovian conditioning and Pavlovian conditioning plus extinction? This question was formed based on previous literature that has studied AMPA receptor changes in the brain, such that AMPA receptor subunits are differentially trafficked onto the postsynaptic membrane in short and long-term memories, spatial memories in the hippocampus. The AMPA receptor is made up of 4 subunits, and there are 4 types of subunits: GluA1, GluA2, GluA3, and GluA4. However, the AMPA receptor is composed of heterodimers, such that the 4 available spots can only be filled with up to 2 different subunits, such as GluA1 and GluA2. There is evidence that for spatial memories in the hippocampus, GluA1, GluA2 heterodimers are trafficked during short-term memory, and GluA2, GluA3 heterodimers are trafficked in long-term memories. For my project, we explored the AMPA receptor in a different brain region, the basolateral amygdala, or the BLA. We chose the BLA because despite it being such a small brain region, previous research shows it's involved in brain circuitry in learning and extinction, as well as reward prediction and motivation. Here, the BLA is shown in a coronal section of a rat brain. Based on this information, after the rats received Pavlovian conditioning, or Pavlovian conditioning and then extinction, they were sacrificed, their brains harvested, and the BLA was microdissected so that the synaptic fraction, which is a section of the brain that is rich in receptors, such as AMPA, could be isolated and analyzed by Western blots to assess the expression of GluA1, GluA2, and GluA3 separately in the BLA. To accomplish this, we used 3 training groups. Our first training group received Pavlovian conditioning and then an extinction protocol. Our second group received Pavlovian conditioning, and our third group received no Pavlovian conditioning. We found that there was a significantly higher expression of GluA3 in our PC group when compared to our no PC group, showing that PC is associated with an increase in GluA3. Our PC plus extinction group was not found to be significantly different from either our PC or no PC group. The asterisk here represents a significant p-value of less than 0.05. Additionally, below the graph are representative Western blots for one animal from each of these groups. A darker band represents a higher concentration of that subunit, such that the darkest band here is in PC, and the second lightest band is in PC plus extinction, and the lightest band with the smallest bar is in our no PC, no Pavlovian conditioning group. For our GluA2 subunit, GluA2 expression was found to be significantly higher in both our Pavlovian conditioning plus extinction and our Pavlovian conditioning group when compared to our no Pavlovian conditioning group. This data suggests that both the paradigms Pavlovian conditioning and Pavlovian conditioning plus extinction traffic GluA2. However, only one of these paradigms, PC plus extinction, had significantly elevated GluA1 expression when compared to both the Pavlovian conditioning and no Pavlovian conditioning groups. This data suggests that the extinction paradigm is associated with an increase in GluA1. This data suggests that Pavlovian conditioning is correlated exclusively with an increase in GluA1, GluA2, GluA3 subunits, but not GluA1, GluA2 subunits, and that the activation of extinction drives the exclusive trafficking of GluA1, GluA2 heterodimers. In our no Pavlovian conditioning group, we found that there was no increase in any AMPA receptor subunit, which suggests that no associations were being made here. Previous research suggests that this differential trafficking in any AMPA receptor subunit during Pavlovian conditioning and extinction may be representing 2 distinct memories and not one being modified, such that there may be 2 distinct cell populations that are active only in Pavlovian conditioning or only in extinction. However, this remains to be confirmed. I would like to thank Hunter College and the Center for Basic and Translational Research, the NIH National Institute of Drug Abuse, the NIH National Institute of Mental Health, our collaborator, Dr. Delamater from Brooklyn College, and the principal investigator and my mentor, Dr. Peter Serrano, as well as the Serrano Lab. Thank you for your time. Anyone have questions? No? Good.
You need to see your notes? No. No, okay. And you are, what's your name? This one? Oh, give me 1 second.
Good afternoon. My name is Sierra Herndon, and today I will be talking to you about a project I had the honor of working on with Professor Greenbaum's lab through the Hunter College SPUR program. Our goal was to characterize the base pairing in the U12-U6 attack small nuclear RNA complex of Arabidopsis through nuclear magnetic resonance spectroscopy. Living organisms are reliant on the flow of genetic information from one generation to the next. This flow of genetic information follows a central dogma where the information of DNA is transcribed into RNA, RNA is translated to proteins, and proteins carry out many of the important biological functions. There's many different types of RNA, including transfer RNA, messenger RNA, and precursor messenger RNA. Precursor messenger RNA includes introns and exons. Introns are non-coding intervening regions, and exons are coding regions that will eventually code for proteins. A large dynamic ribonuclear protein complex, known as a spliceosome, is responsible for splicing or removing the introns, and the exons are ligated. The accuracy of splicing is crucial for gene expression, and inaccuracy